Hello everyone, I'm Mr. Furlong, and today we're talking Mendelian genetics. Before we dive into Mendelian genetics and how it all fits together, I want to quickly review about some common processes that are found in all life forms. We know that shared conserved fundamental processes and features support the concept of common ancestry for all organisms. So for instance, DNA and RNA are the carriers of genetic information. This is true no matter what species you're talking about. Now granted, some species might have RNA and not DNA and vice versa, but most species have both RNA and DNA and they do the same things. We know that ribosomes are found in all life forms, whether you're talking about a bacteria, a plant, a fungus, an animal, us, and they all have the same process. They are the place where proteins are made. We know that the major features of all the genetic code is shared by modern living systems. So for instance, a specific code on the messenger RNA codes for a specific amino acid. This is true no matter what species you're talking about. This is why we can take human DNA and put into a bacteria, and that bacteria is going to read the human DNA and actually make the protein. The core metabolic pathways are conserved across all currently recognized domains. So for example, in the process of cell respiration, where we go through glycolysis, Krebs cycle, oxidative phosphorylation, all those things require enzymes. And those enzymes are pretty much the same in all species. Now what about species that don't have mitochondria? Well, they do have the process of glycolysis, just like we do, and the process works exactly the same way. So let's talk about Mendels Laws. First off, what we have to keep in mind is that this describes the inheritance of genes and traits when they're found on different chromosomes. If the traits are found on the same chromosome, they're not going to follow these rules. We'll take a look at that in another video. First off, a couple definitions. A gene is a segment of DNA that codes for a protein. So we all have genes. For instance, we have a particular gene that tells us what our eye color is. We have a gene that will tell us our blood type. However, an allele is a specific form of that gene. So for that eye color gene, is it a blue eye gene? Is it a green eye gene? Is it a brown eye gene? For blood type, is that the A allele, the B allele, or the O allele? They're all genes for eye color and blood types. They're just different forms of them. So how does this work? An organism inherits alleles from both parents. Now, organisms can inherit different alleles for the same gene. So for instance, mom might donate the A blood type allele and dad could donate the O blood type allele. It's possible that maybe they both donate the A allele. And these alleles are represented by letters. So we use capital letters to represent the dominant alleles and lowercase letters to represent the recessive alleles. Now, how do we get these alleles to begin with? Well, this happens during meiosis, which we talked about in the previous videos. The first person to really study this was Gregor Mendel back in the late 1800s, and he came up with three principles or three laws. One of them is the law of segregation. This says that alleles are found on chromosomes, and that homologous chromosomes carry the alleles for the same trait. So in this example, we have two different chromosomes. One chromosome has the dominant allele for that trait, one has a recessive allele for that trait. When chromosomes are separated during meiosis, well, these alleles get separated as well. So when that cell divides, so do these chromosomes. They end up in different cells. Another one of his laws was the law of independent assortment. This says that two or more alleles will separate independently of one another. Now this will allow for many different combinations of how those traits are going to be packaged in the embryo. And this is why we can do then a Punnett square, because those alleles will be separated and they're also found on different chromosomes. Now this is basically all just probability. So the rules of probability can be applied to analyze the passage of single gene traits from parents to offsprings. So how do we do this? Well, we use a Punnett square. Now you know how to do Punnett squares, so I'm not going to spend time going over that. But you have to look at the wording of the question. So for instance, to determine the probability of A or B happening, you have to add the probability of A to the probability of B. So for example, in pea plants, which is what Mendel studied, yellow pod color is dominant to green pod color. Having round seeds is dominant to wrinkled pea seeds. And if you cross two heterozygous yellow pod colors and two smooth seed pea plants, what is the chance of having either green pods or wrinkled seeds? So we're going to do this by just making a simple 4-box Punnett square. Now both of these are heterozygous. I am going to cross a heterozygous yellow with a heterozygous yellow. So if I do my Punnett square, now I'll do the same thing with my heterozygous round seeds as well. So my question is, what is the chance of having green pods or wrinkled seeds? So my chance of having a green pod is just 1 out of 4. The chance of having a wrinkled seed is 1 out of 4. There should be a 1 right there. And I'm going to add these together. So this is going to be 2 out of 4, or 50%. If I want to determine the probability of A and B happening together, I now need to multiply the probability of A to the probability of B. So in this case, I have a 1 out of 4 chance that it could be green, and a 1 out of 4 chance that it could be wrinkled. And now I multiply it. So now I have a 1 out of 16 chance that both of these happen at the same time. So remember, if it says or, you want to add. If it says and, you want to multiply. We're going to do a lot of practicing with some Punnett squares in class, and I'll see you then.